Morning, everyone. Welcome to our NTLM webinar on change control and validation. So this morning we've got Tim who's going to give us a talk first about change control. Then I'm going to talk a little bit about validation and verification. So I'll hand over to Tim.
Brilliant. Hi, morning everybody. Thanks for joining us. Okay, so my name's Tim Bush. I'm the Blood Transfusion Laboratory Manager at North Bristol Trust in Bristol, so Southmead Hospital. And we're talking about change control. I'm going to be focusing on a blood grouping analyzer, but the whole process applies to all of our change controls. So change control. Yes, so we do change control because it's a tick box exercise we are made to do by MHRA UCAS, where we jump through hoops when we all know it's just common sense. However, that was young Tim back in 2012, and that's what I thought. It's just straightforward common sense. Why do we really need this process? Then I got burned a couple of times, and you realize that common sense is not always common. So change control actually is your best friend. So there's a picture of me with my fashion choice back in 2012 of an awesome moustache. Couldn't grow that now. So what's the aim of this? The aim of this is to understand the MHRA and the and ISO 15189 requirements, risk mitigation. That's why we're doing it as well. Documentation, recorded approach, visibility, change control management. So let's define a change. So change can be anything there's a potential to compromise the safety of patients, staff, or product quality, essentially anything that can affect the validity and the quality management system. So when do you think a change control is required? And also, just as important, when do you think a change control is not required? Now, they seem to change. The lines seem to think. So let's have a look. So for a start, a new analyzer is definitely in keeping with a change control. So any process you sort of got to change, think about doing a change control process. And that can be even things that seem quite small. For instance, MHRA have just changed the SABE reporting system. We should have done a change control for that. So it depends on what change you're doing. It depends how big the document is going to be, how long it's going to take. But the whole thing is it will just control it. So why do we need this change control? It's to ensure that it's planned, it's effective, it's controlled. That's in the title. It's managed, and to ensure the changes will be safe for everyone involved. And also, you can go through the change control process to start with, and it will tell you if you actually need that test. And you might do the impact assessment, which we'll talk about in a minute, and realize actually that change wasn't even required. And it will change, will the change benefit and help with continual improvement? The whole point of doing change is not for the sake of change. We want to do a change because it's going to be beneficial. So what's the advantages? It mitigates the risks to product, patients, and the staff, improves the work lives of staff, cost and time savings, improve compliance with regulatory standards, prevents duplication of work, ensures that it's right first time. It's all that forward thinking, it's all that planning and things like that, just making sure that it's controlled. So what are the first steps, or what the steps to change control? You want to discuss with your quality manager. They are essential to this process. They will think of things you haven't thought of, and they'll offer you advice. Risk and impact assessment. That is one of the really big parts of this. You sit down and you think about it. I'll talk about it in a minute. And you come up with the risks, and then you sort of what the impact that's going to be. Then you plan that change. You identify any validation that needs to be done or used. And then you've got to provide the evidence, record the deviations, and review effectiveness. And that's all part of the process. I don't know if you can see that. It's quite small. But you got a stages. Stage one, so you identify the change as required. So you raise on your CAB system. So we use QPulse. I think a lot of people use QPulse, but there are plenty of other systems out there. So your quality system should have a way of recording this. So we do what's called a wizard. So it just records it. We then do the impact assessment on a form. So you have a particular form. This was actually, I think we borrowed from NHSBT. We saw it, and this was years ago. We haven't changed it much because it seemed to really tick most of the boxes. And as you go on, you can sort of change and add things as you require. Then at stage two, after you've assessed it, is it required? No. Then you might go back to sort of might decide not to do it, but then usually you do. That's the whole point of doing this. Usually is. You then complete your action plan, and then stage three, to do that exercise, you do your sign off, initial go live. So how to do a risk impact assessment. This is what gets people, so it can be a little bit confusing at times, because what we have a habit of doing is running ahead of ourselves. We can see what we have to do. So what can normally happen when you do the impact and risk assessment, you start just writing down what needs to happen, but that's not how we do it. So we assess the risk impact of making the change, the who, the what, the when, the where, and why do it. Use an impact assessment matrix, and you mitigate any risks identified. So here's the criteria. So basically, it's like a checklist, really, if you look at it. So first of all, you think about personnel. What sort of personnel are you going to need? The premises, you talk about the equipment you might need, information technology, materials, consumables, documents and records like your SOPs, all those sort of things. These are sort of things that guide you through the process of thinking about what sort you have to do the changes. As a whole list, you can read those. So how to the risk impact assessment. So you got the criteria, description of the impact of risk, and the action to control it. So for instance, criteria. So one of the sections is all laid out, and you got numbers. We get down to 8.3, temperature control monitoring. Give a description of it and go purchase air conditioning so it does not get too hot or cold. Actions, arrange air conditioning as part of the enabling process. That's not correct. So that's the trap sometimes we fall into, because we know in our mind what the problems are, but we've got to document it a bit better. So the criteria is temperature control monitoring. So actually, you look at it, especially when you're talking about analyzers and also any sort of reagents. If extreme temperature, the lab has got excess 29 degrees before in the past in the summer, that's my particular lab, you may have the same problems. This could affect the analyzer and reagents, resulting in poor quality of results. That's the risk. If the temperature's too hot or too cold, it's going to affect the analyzer. So the risk. So the action there, or what is required to control that risk, is to arrange air conditioning. So that's one of the things a lot of people get mixed up with. They put down the risk as the solution sort of thing. I hope that sort of becomes clear, or is clear. So here's a couple of examples. Staffing levels. Again, a risk. Insufficient staffing levels will mean that the validation and verification will not be done correctly and on time. So insufficient skill mix will also impact the running of the installation. So we're trying to put new analyzers in, and if we don't have staff, that's the risk. That's the risk if we don't have them. We're not going to get it done. So then the action then is to obtain additional support in the form of this comment of band 7 level, or whatever you decide, and that's the action there. Building facilities, here's another interesting one, something that's quite often overlooked. Without the key fundamentals, the process will not work. Sockets are required, waste pumping, etc. So you're thinking about one. I consider little things, but actually the big, massive things. So if you've got nowhere to plug in your analyzer or your bit of equipment, you're dead in the water there. You're not going to go much further. I actually remember a time thinking this, doing it in my head back in 2012, sort of thing, thinking, yeah, we'll just plug it in. But I didn't want to go and check if I had a socket. And when I got there, it's like, I got nothing to plug it into. And it's embarrassing for a start because it's so obvious. But these are the sort of things where this risk and the impact assessment comes into its own, because it guides you through. You're thinking beforehand, getting it all ready. Here's a few more slides. Validation, if not validated, errors may occur. Write validation scripts. There's the actions. Okay, so documentation. User requirement specification. Design qualification based on the URS. That goes to the outline generic validation script. So you got your IQ, your OQ, your PQ. So you got your change control validation policy, change control and validation SOP, impact assessment form, and then new and amend test verification form. So we've done our impact assessment, and if we've done it right, so you've got all the risks and then you've got the mitigations. Those mitigations then become basically your action plan. So how are you going to implement this change? So your change action plan should refer back to risks and impact assessment. And you need to cover the key steps. Set up a date for completion, assign responsible persons, and put in the timelines. If you're aware of QPulse, or maybe your QMS system looks a bit like this, we have a nice way to manage it. So at the top here, you've got the details, what we're trying to do, put the blood group analyze in. We've got an impact assessment. And each of these sort of drop down and they become actions, different areas. So the action plan in particular is one of importance. So you put each one. So you put down who the owner is. Really, you want to spread that up. If you've got your senior staff or BMSs that are helping you, when you put those names in, you've got the details. So, for instance, here, the second one down, update QPulse new analyzer, and the response, you put it in, and you can see here, it's not been done yet. So that's fine. But you've got a timeline. That had to be done by June, 12th of June, 2023. So they'll give you alerts if they ain't done properly. So you've got this little yellow sign up here. And it's a way of just keeping it, I don't know, I suppose keeping it real, so keeping you on track. And you just fill in there. So, and then you just put down, for instance, here, as I was saying, you put the new analyzers in, you put them in, and then you click OK, and it's done. Documentation. So it's the raw data working folders in your QMS software. It might be elsewhere, maybe on your computer somewhere, a folder on the trust drive, etc. And I find that if you put it out in a nice sort of different folders, you can dump all your stuff in there or place all your stuff in there. It keeps it nice and organized. And these are all the other change controls. So as you can see, you've got some open, some closed. It's just a way of controlling it, as we talked about, sort of management. This is from my analyzer, we'll skip over. So I had to redo the whole building. So that's part of it. Then you got the validation stage. I'm not going to talk too much about this because that's what Kerry's going to be talking about. But user qualification, specification, design qualification, you got your IQ, should be performed on new modified facilities, systems and equipment. IQ confirmed the equipment has been installed correctly. Got the operational qualification, establish the equipment is fit for purpose. Completion of successful OQ completion should allow the formal release of the facilities, systems, or equipment. And then you got performance qualification, and that should follow your IQ, OQ, and PQ demonstrates the equipment, system, or process can consistently produce the quality that's required. Don't necessarily have to in that order, but that's how it normally sort of goes. So the evidence and effectiveness. So we need to be able to demonstrate when we come to be inspected or assessed, they're going to want evidence. So you need to demonstrate the plan was followed and all steps were completed. So again, that's where on your QMS, if you've got it all recorded, it's nice. And the important thing is go live is not the end of the change control process. You need to review the effectiveness after a defined time. So for instance, if you've got a new analyzer in, you want to maybe look at it 3 months down the line, or if you've done another change, you might want to look at it in a few weeks to make sure it has been effective. And so you want to monitor that for a month for simple changes or 3 months for more complex changes such as a new analyzer. You want to capture the deviations as nothing ever goes to plan. I don't think I've ever had something that's gone to plan. It seems a little bit too good to be true. So you need to capture those deviations. There's nothing wrong with deviations, Something might happen. Maybe your staff's gone off sick, for instance, and then you have to change the timeline and stuff. Just record it. It's a deviation. Doesn't necessarily mean it's a bad thing. And then evaluation. Look at the error logs. We're talking about analyzers here, but it's this sort of thing. Look at the error logs. Have they been excessive? Repairs, as you can breaking down, need a lot of work. The instance involved in it. Ask your staff, how do you feel it's gone? How are you liking this and stuff? And they might say something like, well, I just don't understand this machine, and you think, oh, okay, have you done enough training? Do we need top-up training? Things like that. Talk about deviations, resource agreed. So this is my personal ones. This is an example of the analyzer. So we agreed extra staff as a secondment, but we weren't able to have them. Something else going on. The MES in hematology required a lot more help than we anticipated. So that was one of the first deviations. That took a lot more staff from my seniors and myself. IT connectivity and setup. We had 3 entities working together, finding time to work together when things had not gone to plan and cost time in verification. So for instance, like I say, it's 3 people. There's Path IT, there's the IT of the analyzers and the IT of our LIM system. Trying to get them together to resolve a little problem was quite difficult sometimes. So that became a bit of a deviation because it put us back. It sort of stopped us processing the a few days or a week. The The other biggest deviation I had was when my staff kept trying to name the analyzers. And even my quality manager got involved, and they, as you can see over here, they still seem to call it Vlad and Mavis. Now that's still on the quality control system. I can't change it. I haven't got access, but there we go. So that was one of my biggest deviations. No, I'm joking. There was more things than that. So to summarize, change control. You have to write a summary report at the closure. So again, this could be a few months after you've actually gone live. So it's got to include the introduction of what the change was and why, a brief description of the validation plan, the methods used to validate the assays or processes, how the results were analyzed, what the acceptance criteria. Then you want to record the sort of results, summary of the results obtained. Not the raw data, that'd just be too big. That'll all be elsewhere, but it's the form of graphs, tablets, and a bit of text. The recommendations. So this should be based on the acceptance criteria stated in the validation plan. So the recommendation was that this was a successful and we should be going live. This change is good. In conclusion, was the validation successful? Basically, actually, I've just repeated myself there. Could anything have been done differently? So again, it's time for reflection. Have a look. Maybe next time do this. And then you have the final sign off. So that's be the lab manager, the quality manager, and the pathology manager. Got different roles. So the quality role and department role is so the quality role, we've got support, advice, provide reference, facilitate meetings, approve stages, be independent, and review effectiveness. And then you've got departmental roles, manage the change, assess the risk, produce the plan, identify stakeholders, do planned actions, review effectiveness. So I've whizzed through that. So the summary is 1, plan. Then you do your risk assessment and you change your plan. Stage 2, act, carry out the change. And then stage 3 is review and check effectiveness. And that's basically the summary of the thought process we go through. But yes, I hope you found that helpful.
So my name's Kerry. I'm a lab manager in Southampton and also the deputy chair of the National Lab Managers Group. So I'm going to talk a bit about validation and verification. So what are we going to cover? Well, validation is a massive topic. So again, we're going to use analyzers as an example, but the principles apply to whatever you're doing. So it might be a new IT system, it might be a new assay, it might be a new premises, a new building. The principles are the same. We talk a little bit about the regulations and why they're there. And then we're going to provide some practical examples for the validation steps and just finish off talking about re-verification. One of the things I would say is, depending on who you're talking to, which regulations you're looking at, whether you're looking at UCAS or the MHRA, the terminology can be a bit confusing and sometimes they use different words that mean the same thing. Essentially, it all means the same thing. What you're doing is validating that whatever your new thing is fit for purpose and does what you expect it to do. You get the outputs that you expect to get. So BSQR says this. 9B says that we must establish and maintain a quality system for hospital blood bank, which is based on the principles of good practice, and that for them validation means the establishment of documented and objective evidence that the particular requirements for the specific thing can be consistently fulfilled. And you'll see these words come up time and time again about objective evidence, and we'll demonstrate some of the ways you can do that later on in the talk. If you go to the good practice guide, it says this. So facilities and equipment need to be qualified prior to implementation, so they should be validated, involving wider consideration beyond the facility and equipment used. So in terms of the good practice guide, validation is a generic sense. That means that you've qualified and validated your activity and it works how you would expect it to. UCAS say something kind of similar, so confirmation of plausibility for a specific intended use or application through the provision of objective evidence, and BSQR talks about objective evidence. So why? I'm probably similar to Tim. If you asked me 15 years ago, I would have said, oh, this is a real pain in the butt, this is bureaucracy, we haven't got time for this, but actually, if you don't do it, there will be times when you will get burned. And the bottom line is this is to keep our patients and our staff safe. If you don't validate whatever it is you're using, in this case an analyzer, and it doesn't work in the way that you expect it to, you could be giving unsafe or incorrect results for your patients, which obviously in transfusion could have massive implications for their safety. So we want to make sure that we're producing reliable and repeatable results, and that we're able to identify any problems, whether they be errors, malfunctions, or issues within the system that you're validating, in the context of your own lab. So that's really important. PQ is the context of your own lab, your own environment, your own staff and personnel, and your own workload. So the sort of tests that you're running. You have to control the process, and this is detailed in the requirements. So within your laboratory or QMS, you should have a strategic document, which is your validation policy, which is your overarching, how do you do this? How do you do validation? And then for every validation you do, you should have a master plan, which details out the critical processes for that validation. And that's an operational document. So that's the practical, how are you going to do this thing? Your validation plan should really include this list of things. So roles and responsibilities, who's going to do this, who's responsible for what, the reason that you're doing it, what's the strategy for doing it, why are you doing it, and how are you going to do it, the time frame that you expect it to be done in. If you are using equipment and reagents, what's their status at the time? So you should be using validated equipment to validate a new piece of equipment. So you've got that kind of traceability through. What are you going to test, and what are your planned acceptance criteria? So that's really important. So you need to plan up front, what would I accept? It may be that the final outcome is that that's different to what you originally planned, but you should have an acceptance criteria at the start, not just run some stuff and then at the end go, oh, I think that's okay. So you should be planning from the start, what are we expecting to see here? We talked about deviations in change control. It's the same in validation, so you should document any deviations to your plan. And this is linked to your change control, as Tim was saying, so you should reference that within your validation plan. And at the various stages, you should have appropriate sign-off. So a sign-off at the beginning that says the plan is good and this is what we're going to do. There may be an interim sign-off if you partially go live before you completely finish. Sometimes that's acceptable, that's relevant. And a final sign-off that says, yep, everything is okay and we're good to go. Or not, as the case may be. Steps in validation. So Tim touched on this. So your IQ, your installation qualification, usually performed by the supplier, whatever it is, whether it's an analyzer or an IT system or whatever. And what that does is ensure that the system has been installed in accordance with the manufacturer's requirements. The next step's an operational qualification or an OQ. Again, usually the supplier. And that checks that the system operates as expected from their point of view. So if it says the heat block must be 37 degrees, that the heat block actually is 37 degrees within their expected range, and so on. The part that's really important to us, and the part that should be done by the laboratory, sometimes you'll get companies offering to do the PQ or the performance qualification for you. That's not really a good idea, because this is about you and your lab and how it works in your system. So the PQ should be done by the laboratory team. And that verifies that your user requirements are met. So you will have had a URS at the start. So the URS is part of your plan. The things that you wanted in your system, does it actually do that in real time? You should be challenging the functions and thinking about your environment and a range of parameters. You're probably in real life going to have different operators, different times of day. So make sure your validation reflects that. So what we're doing is a collection of tests that verify the system performs as expected in a simulated real-world condition. And we've talked about should be done by the customer, so it's non-biased. So if you put those three things together, what you get is an IQ that says everything's been installed correctly, an OQ that tells you physically it's operating correctly within its limits, and the PQ tells you that it gives you the right result and does the right thing and it's safe and consistent. So we're going to use some practical examples. Again, as I said, we're focusing on an analyzer, but imagine this could be anything. It's just what you would be testing would be different if it were an IT system or a room, for example. So with an analyzer, what we'd suggest is a sample or result comparison with your current validated system. So you're going to have an analyzer that you're using currently and a new one that you're bringing in. Suggestion is we would run in parallel for a defined time period. That might be a week, it might be a day, it might be 3 months. It depends on your workload and your decision in your original validation plan. You need to select a number of tests that you're going to do, and that's a risk-based decision. And one of the questions we're often asked is, how many should I do? And that depends on your workload, your normal process. How many do you see usually? You take a percentage. There are some sort of statistical calculators out there that can help with this. There's a UCAS link that I can put in the chat later where you put in how many of these things you would normally do, and then it makes a suggestion as to how many you should do to validate a system. You should challenge the system with things that are more complicated or maybe you don't see as often. So when we're talking about blood grouping or antibody screening, there's some suggestions there about weak Ds, weak reverse groups, weak antibody screens, variants. Cover all the possible outcomes that you're going to see. And remember, it is about your requirements and your service. So what is relevant to you? There may be something in particular that you do that's quite special that maybe other places don't do. Make sure you cover that in your validation plan. Or maybe you never ever see any of these things and then it might not be appropriate or applicable to you. You should be looking at reproducibility, and there are some different ways you can do that. You can run the same sample multiple times. That's repeatability, really. Do you get the same result if you run it multiple times? What I'd really suggest is you should run it multiple times over a period of time, bringing in those variables that we were talking about. So different operators, different times of day, and then that you can also bring in reagent stability if you do it that way. You should be looking at sensitivity or detection limits. So one of the ways you can do that is to use a metrologically traceable anti-D. There are plenty of them on the market to check that you are detecting the limit that's in the compatibility guidelines. You can look at EQA across all the platforms. And remember, it doesn't end with just the analyzer. There is interface validation as well for connectivity, and that, as I said, IT systems, it's the same thing. What do you want it to do? Test each of the variables for how it will do that and challenge it with some of the harder functions. And then UCAS, they love uncertainty of measurement, but it does have a use in validation. So it's a measure of success. So if in your plan you say, I expect my uncertainty of measurement to be 95%, that tells you that 95% of the time you're getting exactly what you expected to come out. So it's another acceptance criteria that you can use. So I said we were going to use some practical examples. So this is a plan. So this is a predefined acceptance criteria before anything's actually been done. So you've thought about what are the things I need. I've looked at my URS. I've said what I wanted it to be able to do in the first place. And I've thought about the technical side. So there's some things here that may have come out of your URS. So as an alert to show that the maintenance is required, the acceptance criteria, as it conforms, it does that. I get the correct grading reactions. It conforms with my existing validated method within plus or minus 1 of the current. So what you're saying is this is what I want to happen, and this is how I'm going to accept that that is correct or it works as I expect it to. When you come to actual tests, running numbers of tests, I said risk-based decision. So an example of that is the first brand new analyzer, new to the market, never had it before, it's new to your department, no one else has got it maybe. You're probably going to want to run a much bigger number, and you might do a whole week's in parallel. I've put an example of 500 samples tested in parallel for a blood group there. If you're moving on and you've got a managed service contract and you've bought in that first brand new analyzer that's never been seen before, and you're going to have a second, maybe a third one, you can start to pare down those numbers. You've covered the highest risk of this is a whole new technology to us, so you can start to bring down the numbers. And then you might get to a point where you're just having a model update. So it's essentially the same with some minor differences. And you can see then we're coming down into maybe do 20 groups and screens in parallel. And there's some examples here of the different things that you might want to be looking at. So antibody screens, baby blood groups, confirmed groups, cross-matching. You need to make sure you very clearly record the results that you obtain and that you record them in such a way that someone who's looking at this down the line would be able to understand what you've done and link that back to your evidence. So objective evidence came up a lot in the regulations, in the terminology that we were looking at. This is where the objective evidence comes in. And it may be an auditor that comes further down the line. It might be MHRA coming in to see how you've validated your new limbs and want to see the evidence that's linked to that. So you can see here, we've got our original, what did we expect it to do? What was our acceptance criteria? Did it pass or fail that acceptance criteria? And how would you know that this was really done? So one of the things an auditor might say to you is, well, you could write this first 3 columns with never having actually, how do I know that you've actually done this? So you need to have some objective evidence to prove that you did whatever you said you did. Tim mentioned before, things don't always go to plan. It might not quite do what you want it to do, but actually that may be okay for you. You might decide that it didn't do what I expected it to do, but how it works is fine for our service. So you would record that as a deviation. So you don't change your original acceptance criteria. You have what you expected it to do, and then you have what terminology is usually a deviation pass. So there's an example here. We said we expect there to be an alert when the card waste box needs emptying, and we expected it to conform with that. But actually, because the analyzer shuts down and clears the memory, it empties on every shutdown, and so it automatically does that without an alert. So the mitigations and make sure staff are aware, put in an SOP. We've accepted a deviation pass. It didn't do what we expected it to do, but what it does do is fine. Therefore, that's okay. Of course, you may have a deviation that isn't acceptable, and that's when you're not going to sign off your validation, and you're going to have a conversation with the supplier as to how you're going to get around that. So if you have a deviation, you need to understand and explain that deviation as part of your validation documentation. So you're going to say, wanted it to do this, it doesn't do this, it does that, but that's okay because. So risk assess, is it acceptable and safe? Mitigate, is there anything you need to do to mitigate the fact that it doesn't do what you expected it to do originally? Do you need some extra training? Do you need a line in your SOP that tells people what to do? Do you need to change your current procedure to fit in with how this new system that you're validating is going to work? And always document. Put it in your validation plan. I'd suggest you put it in as a non-conformance as part of your quality management system as well, but make sure it's clearly documented what you did and why. And as I said, if it's not safe, it's not acceptable, you can't mitigate it, your validation has failed, your user requirement hasn't been met, and you're going to need to engage with your supplier, and you're not going to put that piece of equipment into practice. We talked about evidence. These are some practical examples of the sort of evidence you might keep. So that this is a spreadsheet of numbers and what was tested and what the result was on the validated system and what it was on the new system and whether that's acceptable or not. Right down to actual images from the analyzer in question in this case, so a raw output, and you link that to your master plan. So if someone says, show me how you compared 20 blood groups from one platform to the other, you would be able to pull this up and say, here is my evidence, this is how I did it. So moving away slightly from the original validation, this is something that causes some confusion sometimes for people. Verification or requalification, depending on what you're reading, or they'll use different terminology. So this is the thing that's been validated once and you've had it in use for 20 years. And I think, years ago, we just assumed it worked for 20 years, but there's recognition that things change. And sometimes the piece of equipment or IT system or whatever it is isn't actually fit for purpose anymore. So re-verification will pick that up. So the good practice guide says that equipment, facilities and systems should be evaluated at an appropriate frequency to confirm that they remain in a state of control. You can't just validate once, you've got to check periodically, and that period is definable by you, that it still does what you expect it to. It's not the same level of as it was with the original validation, so you're not going to be running 500 samples, for example, unless you think you've got a problem, then you might want to. So what you're doing is verifying that status hasn't changed. The analyzer in this circumstance is still fit for purpose as the environment has changed around it, i.e. your workload, the building that's around it, your staffing, whatever happens to be happening in your service. How do you do it? So I would say use a risk-based time period. We don't want to make ourselves too much work here, maybe every 2 years, maybe every 3 years, but you can justify that with a risk assessment as to why you think that's an acceptable time period. And you'd be bringing in things like we run IQC, we run EQA, we know that our analyzer essentially works as we expect it to. It should be planned in the same way as a normal validation is planned, with a validation master plan, but likely to be a much shortened version. It won't include an IQ/OQ because you're not installing or operationally qualifying a new thing. This is just making sure that it still does what you expect it to do. It may or may not include practical testing. So you might decide that you want to do some practical testing, but you might decide actually, if I review my IQC, my EQA, and any instance and downtime I've had, that's enough to prove that this system still meets my expectations. And you would document in the same way in your plan with an acceptance criteria and whether it passed or not. So just some references there for you, some useful BSH guidelines on validation and qualification include a lot of this stuff, and change control, really useful tool there. This is the newest. We had some confusion, I think, over the good practice guidelines. A lot of people were using the wrong one. So there is a new 21st edition, actually came out in 2023. It's linked to the EMEA. So make sure you're using the right version. They're not wildly different, but there are some differences. And of course, we're now on the 2022 ISO standards rather than 2012.